Dear students, today I'll be speaking on cell membrane and its introduction. After the invention of electron microscope in 1950, the research activities in the field of cytology paced up a bit, and many a hidden theories came to light because of this invention. To study plasma membrane, RBC plasma membrane is considered as the best representative. Plasma membrane is mainly composed of lipids, proteins, and small amount of carbohydrates. The major lipids are phospholipids arranged in a bilayer like this. This is the lipid bilayer number 1 and this is lipid bilayer number 2, and the phospholipid molecules contain head and tail. The phospholipid molecules are polar in nature. That means they are amphipathic. They have polar heads which incline towards the watery ends, and they have hydrophobic end which inclines themselves toward the inner side of the plasma membrane. These are non-polar tails of saturated hydrocarbons. They are protected from aqueous environment because of this amphipathic nature of the phospholipid molecules. The main phospholipid components are phosphoglycerides, cholesterols, and apart from these two, the membrane also contains proteins and carbohydrates. The ratio of protein to carbohydrates is variable and it is not fixed. We have around 52% of proteins and 40% of lipid in the plasma membrane. The proteins which are embedded in the plasma membrane in the lipid bilayer, they are of two types, and they are classified on the basis of ease of extraction from the plasma membrane. The peripheral proteins, these are the proteins which are very easy to extract and they are majorly laid on the surface of the membrane. Apart from peripheral proteins, we have integral membrane proteins. They are difficult to extract and they are partially or totally buried in the plasma membrane. These proteins also contain small oligosaccharides or sugars upon them. These make the glycoproteins, and when the carbohydrates they are present on the lipids, they are called as glycolipids. These are the peripheral proteins which are on the outside of the on the peripheral part of the plasma membrane. These are the heads of the phospholipid molecules like this. This is the head and this is hydrophobic tail. This is hydrophobic tail and this is hydrophilic head. Hydrophilic means it is water loving and hydrophobic means it doesn't get attracted towards the water molecules. These are the cholesterol molecules which are present inside the plasma membrane, and they are the membrane stabilizers. Now there are some membrane labilizers also which confers the fluidity to the membrane, whereas cholesterol molecules they confers rigidity to the molecules. The best accepted model of the plasma membrane is fluid mosaic model, which was given by Singer and Nicholson. The broad outline of the fluid mosaic model, we will be studying the fluid mosaic model in details in the upcoming lectures, but here I would like to tell you about the outlines or the broader aspect of the this membrane model, which was put forth by Singer and Nicholson. The membrane here is quasi-fluid, means it is not totally rigid. Rigid molecule doesn't explain rigid membrane doesn't explain the fluidity of membrane. That's why the best conceived model is fluid mosaic model. The quasi-fluid nature permits the movement of proteins within the bilayer. This movement of protein enables the plasma membrane to flow and to merge into another membranes. The ability to move within the membrane is its fluidity, as I've told you. Fluidity helps in cell growth, formation of intracellular junctions, secretions, endocytosis and exocytosis, and cell division. Had the membrane been non-fluidic, all those aspects which I have mentioned right now could not have been possible. Another peculiarity or characteristic feature of the plasma membrane is selective permeability. Selective permeability means it is differentially permeable. The membrane is differentially permeable, means it may be permeable to some, it may not permit some to come inside or go outside. The transport are of many kinds. Passive transport is the transport in which energy molecules are not expended and they are not driven by the energy. Such a transport doesn't require energy. The simple diffusion is the process of transport across the membrane in which neutral solute molecules moves across the membrane along the concentration gradient. Along the concentration gradient means higher to lower. Movement would be caused from higher concentration to lower concentration. Okay, then osmosis. Osmosis is the movement of water across the plasma membrane by diffusion, and the water molecules move across from higher to lower concentration. Another type of transport is facilitated transport. The molecules which could which are non-polar in nature can easily cross the non-polar lipid layer. However, those which have polar characteristics, they may not be able to cross the lipid bilayer. That's why they have to be ported through special kinds of proteins called as porins, and these porins affects the facilitated transport of polar molecules. These polar molecules, since they are hydrophilic, cannot pass through the non-polar hydrophobic bilipid layer. In this case, we require a carrier protein of the membrane to facilitate the transport of polar molecules. Like this, these are the solute molecules. and they have to be ported inside. For this is the carrier protein. They change their conformation and provides the space for solute molecules, and by change in conformation they port the solute molecules present outside the plasma membrane inside the ICF or intracellular fluid. The most important of the transport is active transport. This active transport involves sodium potassium pump. When I'll be taking plasma membrane in detail, at that time we will discuss what is sodium potassium pump. Right now, active transport is the transport of solute molecules from lower concentration to higher concentration, that is the movement against the concentration gradient. Such a transport from lower concentration to higher concentration requires expenditure of energy molecules, or it is driven by ATP. This is all about the introduction of the plasma membrane. Let us move to another organelle called as ribosome. I have told you at the initial stage of this series that ribosomes are the membrane and are the membraneless organelles. They are present in both prokaryotes and eukaryotes. First of all, they were reported by George Palade in 1953. This was the first structure which was observed under the electron microscope. No membrane surrounds it. If you could remember or reminisce the nucleus, you find some darkly stained areas which you refer as nucleolus. This nucleolus is nothing but the site of rDNA which get transcribed to rRNA, and it gets associated with some proteins and forms granules called as ribonucleoprotein particles. These, how ribosomes are formed? From rDNA upon transcription it forms rRNA plus proteins forms RNP particles. These are nothing but ribosomes. Okay, these are ribosomes. Now, the eukaryotic ribosomes, as I have already told you, they are of 80S type. They have two subunits. One is 60S and another is smaller one is 40S type. The prokaryotic ribosomes, they have largest subunit as 50S and smaller subunit as 30S. S stands for the Swedberg unit. I have told you in the last lecture. It is the sedimentation coefficient. And organelle ribosomes, which is the characteristic of two organelles. One is mitochondria, another is chloroplast. The organelle ribosomes are of 80 Swedberg unit type. Because these organelles they were originated by the endosymbiotic relationship of eukaryotic cell with bacteria, they are of 70S type. Okay, so this is all about the two organelles I have talked about today were plasma membrane. In fact, plasma membrane is not considered as an organelle, but it is very much the part of protoplasm. And one organelle we have discussed right now in short is ribosome. In the upcoming lecture, we'll be dealing with introduction of mitochondria, chloroplast, and other such organelles. Thank you.